The work that we do in the Institute in general is to look for any genetic or environmental intervention that extends healthy lifespan. Everyone knows that if you eat badly, you live for a relatively short amount of time, and there are all sorts of side effects associated with that. And the same is true for the organisms that we use in the lab. We use flies, and if you give them a bad diet or an imbalanced diet, they'll live for a relatively short amount of time. The Drosophila melanogaster, which is the species we use, they're called fruit flies, and so they feed on rotting fruit. So the fungus grows on the fruit as it rots, and the majority of the protein and vitamins that they get in their diet are from the fungus and the yeast, fungus that's growing on the fruit, and then they'll get some sugar from the fruit itself. So it's this mixture of sugar and yeast that we try to reproduce in the lab by feeding them a sugar yeast diet. What we find is that we can manipulate the yeast in very subtle ways to modulate lifespan. One of the more important things we do is, and we're focused on, and what we found for the effect on lifespan at least, is that when you manipulate the ratio of protein to carbohydrate, it has a very big effect on fly lifespan. We're looking for ways that this could be applicable to humans. So hopefully, yeah, these dietary interventions will be useful for humans. And then also the information we learn about the molecular pathways that they access will also be useful for improving the health of humans, so that we can come in with dietary interventions that are not whole scale but that nutrient specific or drugs. So my lab at the moment contains a postdoc, a technician, a couple of PhD students, and another lab assistant. The way that we work together at the moment is we have a weekly meeting at which each person in the group presents what they've been doing for the last week and what their plans are for the following week. And everyone has a chance to input then on what people are doing at the technical level through to the scientific level. The postdoc in the lab at the moment, he's working on the interaction between very specific nutrient changes and a drug called rapamycin, which affects a molecule which senses protein inside the cells. And so the interaction between this drug rapamycin and lifespan and those amino acid or those nutritional components. PhD student Sahar is working on the details of the molecular pathways that sense those amino acids. So there are at least 2 of these that we're working on at the moment. And then what happens when those amino acids are sensed is that they start a trigger of transcription and translational changes, so changes in gene expression that then knock on to affect the organisms. I also have a PhD student who's interested in what happens at the physical level in terms of changes with flies. Flies are great because they're so small and they live for a short amount of time, but at exactly the same time, this is one of their Achilles heels essentially, because they're so small that we can barely tell whether a young fly from an old fly. But one way that we can tell the difference between old and young flies is that they move differently. In particular, if you ever watch a young and an old fly next to each other, you'll see that one falls over a lot, one is slower than the other one, it stands still for a long time, it has worse sleeping patterns than the younger fly. And it's very obvious. So we hope to be able to use video-based methods for diagnosing ages in flies. My role in particular, I see it as trying to stay on top of the broader literature and see what's interesting and important for us to be pursuing, and also to make sure we coordinate our activities with the other groups in the IHA, which there's a worm group here and a yeast group, as well as a rodent group that we interact with. And we need to be coordinating our activities all the time so that we're working on research that's relevant and that's evolutionarily conserved between the modern organisms. And we're not just developing a fly-specific aging project that's completely irrelevant and not of any interest to anyone.